Hello everyone. Previously we talked about global sequence alignment and how we can use dynamic programming for it. And today we will focus more on local sequence alignment and how we can use dynamic programming for this one. Let's see what are the differences. Let me bring this slide. Yes. So in order to compare it, let's go through it one by one. So these two are exactly the same. So score of aligning A and B, still we have it inside the definition. And we have also optimal similarity of these two sequences. Now, take a look at the relations. Now we have some differences. For the first row and first column, you can see that we assign all of them as 0. So if you have such a table, you see the first row and first column, all of them are assigned to be 0. It was not the case for global alignment. So this is the first difference. The second one is related to this 0 inside this Fij calculation. The three components that we had before, they are exactly the same, no changes there. But we also added 0. What does it mean? It means that when you are calculating the maximum, if all of the values are negative or equal to 0, then at the end you will have 0 as the final answer. It means that inside your table you won't have any negative value. But what's the goal here? Why we are doing this? Because the series of these zeros will make the things much easier for us to see the trend and try to find the optimum answers much easier. So that's the goal for local alignment. And we can jump from one place to another, and I already showed you in one of the slides before. So let's take a look at this table as an example and see how we can find the possible path for our alignment. Now, for the global sequence alignment, we always use this value at the end of the table and try to trace back somehow into the first value, right? But in the local alignment, it's not necessarily this way. One way is to find the maximum value in your table. And if you take a look at the whole table, you will find out that 14 has the maximum value. You may say, okay, maybe I will have another 14. Yes, maybe that one would be another possibility for you. So I choose the maximum, for example, and I try to trace back. So when you are trying to trace back, not necessarily you will reach the first cell, because in global alignment, you always start as your initial optimum answer to the first cell that you had inside your table, not in local sequence alignment. So in this case, let's see how we trace back. So we have the maximum value inside the table. Again, take a look at your neighbors. This one has a bigger value comparing to 9. But take a look at this one. E aligns with E. Maybe this is a good path, right? So not always we choose the maximum neighborhood. Sometimes we should think, for example, these two are good alignment together. They are a match. So it will make positive. If I choose this path, then again, I will have another mismatch here, another mismatch here. So that's not a good idea. Sometimes it can be tricky. Usually we try to choose diagonal as much as possible, but sometimes having a gap can be very helpful. Okay, so for this one, this is maximum, so no question, we go here. And this one and this one has the same value. We can think about it, which one to take. Usually diagonal is better if this one doesn't give us any advantage. And we continue until here. Finally, we are surrounded by zeros. So you see, these zeros are very helpful for us to understand where to stop. Then you don't need to have terminal gaps before it so that you reach the initial point. You don't need to do that. So this path could be a possible path for our optimum answer. So why I always say possible, because it's not 100%, it's not guaranteed that this one is the best, but this could be one option. So as you can see, such tables can be useful for us. So instead of considering all the possibilities for comparing these two sequences together, which are very short, assume that they are very long, so it will be very tricky. And tables can be very useful for us. So some possible local alignments inside the same table that I showed you here. Now you see, for example, assume that you chose this path exactly that I showed with green font. So in that case, F aligns with F, E aligns with E, L with L, A with V, so there is a mismatch, but we align them anyway, and K with K. We have only one mismatch, and all the other four ones are matched. This one, see? So in order to write these sequences, it's important to know how we can write them from the table. When we have diagonal like this, it's very easy to understand. Like I have K, A, L, E, F. Without tracing back, be careful. When you write the sequence, it is from the left to the right, and also here from the up to the down. Okay, we should be careful about that. So K, A, L, E, F. You see? Now assume that we have a gap. Which path did we choose here? Now, what was it? K, A dash. K, A dash. What does it mean? So we had this path, and from here we had a dash. Instead of going to the L1, we have a dash. It means that we go down. So this is the path that we have chosen. So instead of moving on to L, we are just going down, just applying a gap here. You see, that's how we can understand how to transform our table to sequence and sequence to table. And after that, we don't have any gaps. It means that we move on to the next cell and we reach 14. That's what happened here. See, KA gap. And again, we went back to the diagonal and we continue. Even from here, I could say the same thing. Let's say, for example, K gap V. Which path is it? So we have K. Then we will have gap. It means that I don't go down. So it means that I go to the right side, for example. Let's take a look again. Yeah, this one. So we can even consider it from up and down. But my suggestion is to just take a look at your first sequence and use that one, because it can be confusing, because sometimes you may make a mistake. So choose another number by mistake instead of the other one. So when you choose only one sequence, so for sure, you know this is the path that you are taking, not this one, for example. Okay, so it would be better. Like for example, the next one. Now we have K gap A. Let's see how we can show it here. So we will have K gap. It means that we don't go to A. We go 3, then 2. So it needs a little practice to see how we can do that. But as I suggested, you just fix it for one sequence instead of doing it for two alignments together. It could be confusing. If you fix it for one, for sure, it's very easy for you to understand which path. So for example, if I have this green color, it's very clear that we are dealing with just diagonal case. Now, as soon as we have such alignments, that's the question to ask. Which alignment is better? So inside your first assignment, you have seen that how we can calculate these things. And sometimes for gaps, we don't give you, for example, any difference between new or extended version. We can penalize them by the same value. It's possible. But sometimes, which is more meaningful, we can apply different penalties for different gap types. take a look at this example, and I want to show you why local alignment can be useful. Like, assume that we are dealing with beta-globin. So beta-globin gene contains a sequence like other genes, and also we can consider the whole DNA. When we talk about the whole DNA, it means that it contains even the non-coding regions, or the junk region as they call it. But you know, even before exon 1, because I already showed you one slide, it contains exons, introns inside the gene. There is a region before even starting exon 1. That's the first place that we are synthesizing amino acids from it, okay? So before that, there is a region which is important. We call it promoter region. This is not related to synthesizing protein directly, if I would say, but it's very important because sometimes you can see some similarities which is meaningful, and it shows that maybe indirectly it has some functions here, and if we change the codes here, maybe there would be some problems in synthesizing the protein, which shouldn't be, but it is. That's why this part of DNA, we call it protein region, which happens to be before exon 1. And we show them with negative values as their position. So when we have 162 and we have negative value, it means that this is inside the promoter region before even we start those codons, those 3 nucleotides that we talked about before, okay? So that's how we can show them. And starting from exon 1, we have position 1, and we continue. So inside the promoter region for beta-globin, for different organisms, like for human and gorilla, you can see this position exactly has the same code, you see? And sometimes there are similarities, but not, for example, 100% the same. That's how we understand there should be maybe a function that they are responsible for. That's why such local sequence alignment can be very useful for us when you find out such similarities, okay? And how we understand that, for example, a similarity is meaningful, because it's not always 100%. And in that case, next week, I will talk about statistical significance, and that part is very important to shape the way that we are comparing such sequence alignments with each other. And that's why next week session would be very important, and we can talk about it for sure next week. So very briefly, I will talk about heuristic technique. This technique is any approach to problem solving or self-discovery that employs a practical method that is not guaranteed to be optimal, but is nevertheless sufficient for reaching an immediate short-term goal or approximation. So in this method, we have approximation, and that's the same thing we have for a statistic. You can see lots of approximation. Personally, this is my favorite technique, because many systems that we have in the nature, it's not solid as mathematics, you know. In mathematics, we try to show everything by logic and also using theorems, and little by little, we are building the whole model. But in heuristic technique, sometimes you should guess things. Sometimes you should approximate. And the way you approximate also is very important, because you cannot just guess, for example, something without any logic. So in a statistic, for example, we say 95%, I'm confident that my value should be inside this interval. This is an approximation, and this one even has a percentage related to that. So these things also are somehow accurate, and the methods that we are using are very important, which one we should choose for a specific problem solving. And what you should know about heuristic technique is that it is not guaranteed to be optimal, because it has errors. Not necessarily the answer that you will find will be the correct one to make it optimal. But the thing is that it shows us the way. In the place that we cannot model everything correctly 100%, this kind of techniques can be very useful. So when I say why do we need such techniques, it's because sometimes we cannot find the answer 100%, and there are some reasons for that, like for example time problem. We don't have all the times in the world to solve a problem. For example, let's say you want to know what's the average height of students in Metro. So one by one, you should go and ask them, or ask them to fill a form. So this thing is time consuming, but assume that you guys are in one class and I ask you separately for example, 30 number of students. So I get ideas that, for example, what would be the average height of you for Metro. And that's why when we are talking about very large sequences, it's very time consuming to consider each nucleotide with another one and for all organisms that we have in the nature. So you see, time is a big problem, and in order to save time, we can use such heuristic techniques to avoid some unnecessarily options. Exactly the way we are doing in dynamic programming. In dynamic programming, I showed you in pictures that we use a path. Not 100% this is the best answer, but at least it shows us some possible path which could be useful. So that's why heuristic techniques can be useful in order to save time. And space problem, it means that for each one of the nucleotide, you need 2 bits in your computer, because we are not dealing just with 0 and 1s, we are dealing with 4 different options. And assume that when we have amino acids, it's even bigger. So the space problem is also another issue. You cannot just compare all the sequences in the world using your computer in a specific time. That's why heuristic techniques can be useful in order to overcome that. And as an example, class that we introduced before uses heuristic techniques for sequence alignment. And for seeing this in practice, I have uploaded one video on O2 class for next week. I suggest you guys to watch it after we teach you about statistical analysis, or even just the term expected value, if I may, because that term is very important in order to understand about this database better. So let's first let me teach you about the statistical significance of sequence alignment, and you will be ready to watch that video. The video is optional for you. I'm not going to ask this on exams, but I really recommend you to watch it. They explain it very good, that how we can use BLAST in order to find some useful information, and maybe even later in our course we will use that to find out about some similarities between 2 sequences and even use those expected values. Okay, so it's very good to watch that one for sure next week. So okay guys, it was for today, and if you have any questions, please let me know. Otherwise, I will see you guys on Monday.